All right, welcome to our podcast on bacteria. I'm going to try to divide this up into 3 short segments, so let's get started. All right, characteristics of bacteria. Bacteria, as you already know, are the simplest life forms we have on the planet, and all bacteria, of course, are prokaryotic. That is a must-know thing. They have no organelles. They have no nucleus. They do have ribosomes, but they aren't really a membrane-bound organelle. There's little clumps of protein, so they're kind of different, but no compartments, no compartments. They are the smallest living things, the very simplest living things. Remember, viruses are simpler, but they're not living. And they have a single circular chromosome. So when we're talking eukaryotic chromosomes, we're talking about a linear, wow, that's weird, a linear DNA molecule that is a double helix like we've always talked about. What we are talking about with a bacterial cell is a piece of DNA that is in the shape of a circle. It's still a double helix, okay? It's still a double helix, but it's in the shape of a circle, okay? And of course not contained in the nucleus. Bacteria historically have been classified by how they are shaped. There are many other characteristics that go into it now, but some of the common language is centered around shape. So, and how they are arranged. So shape, language terms associated with shape. If a bacteria is shaped like a sphere, it is called coccus. If it's shaped like a rod, kind of like this or like this, even better, it's called bacillus. And spiral, like this one over here, it's called spirilla, okay? They're also classified by how they're arranged. Now we know that they're always unicellular, but they can hang out in little groupings. If a particular species of bacteria tends to hang out in pairs, little pairs of 2, and then it's referred to as being diplo. If it hangs out in bunches, like little bunches of grapes, it's called staphylo. If it hangs out in groups that are in a line, it's called strepto. So this might ring a bell if you have ever had strep throat. Streptococcus bacteria is what has been causing that most of the time. Strep throat is a chain of sphere-shaped coccus bacteria. Okay, so that's how that naming works. And of course, there are many more complex names than that, but it's a historically useful terminology. All right, structure. How are bacteria built? Okay. What you need to do on your paper or on some paper somewhere is make a drawing. I'm going to make my drawing up here. Okay. And that portion of the bacteria that I just drew is the cell membrane, which of course controls what enters and leaves the cell. It's made of phospholipids and all that. Okay. Now, around the cell membrane is always in bacteria a cell wall. So I'm going to draw a cell wall next around that bacteria. So cell wall, and we know from our notes about the kingdoms the other day that the cell wall is always made of something called peptidoglycan. It's a protein carbohydrate complex, but it helps the thing maintain its structure. Now, a third layer around many bacteria, though not all of them, is something called a capsule. And so I'm going to kind of draw that third layer around it. Okay. And it's kind of sticky and allows a bacterium to stick to some kind of substrate surface and not be just washed away. Okay. There is another set of structures called pili, and we can see the pili in this bacteria, these strands of almost hair-like looking things. So these pili, which stick out from the bacteria. They kind of look like cilia, if you ever remember learning about cilia before, but they're not. Cilia help a structure move, and these don't move. These things just basically act like Velcro. You can think of it as bacterial Velcro, and again, helps them to stick to a substrate or to other bacteria. Okay. Finally, some, but not all, bacteria have a flagellum, and that flagellum is a long bundle of microtubules, which is just a skeletal structure that allows, when it's moved back and forth, it allows the bacteria to weakly swim through a fluid. And finally, DNA have genetic material. They have that single circular chromosome we were talking about earlier, okay? And basically, that's it. That's their DNA. This all through here is cytoplasm. Sometimes bacteria also have little smaller extra bonus DNA molecules that look just like that one, only smaller and have very few genes on them. Those things are called plasmids. Whoops, so that was poor planning. MIDS. We'll talk about those more in a minute. Oh, and that is a picture of a flagellum right there. You can see that flagellum. Okay. All right. Next, reproduction. This is what bacteria are all about. Well, it's what any living thing is all about, but bacteria are especially good at it. Now, remember, bacteria do not have linear chromosomes like we do, that we are used to seeing in pairs and all that. And just they do not, there is no mitosis, there is no meiosis in bacteria. They don't do that. They don't have to organize their chromosomes and so on. They do something that superficially to us would look a lot like mitosis or meiosis because it results in the cell pinching apart. But there is no organization of chromosomes, so it's a very different thing. All bacteria reproduce by asexual reproduction. So if you are a bacterium, there will be no sex for you. It is all asexual. They are cloning themselves. Binary fission is the method by which they basically copy themselves and split the cell in half. So it's a very simple process compared to mitosis or meiosis. The chromosome, that circular chromosome, is simply copied. Then the two circular chromosomes get separated to opposite ends of the cell, and then the cell pinches in half. It is a really super straightforward process. Binary fission. Reproduction, because that process is so pretty much straightforward, is really rapid. Every 20 minutes, bacteria can reproduce. So if there were unlimited resources, you can imagine that you could grow an enormous bacteria population pretty quickly. 1 becomes 2, 2 become 4, 4 become 8, yeah, Coleman, good. 8 becomes 16, and so on and so on and so on, okay? So you could get that duplication happening really fast. It is limited resources on the planet that keep bacteria from growing out of control. But if a strain arises that can take advantage of some unique environmental situation, like maybe it is resistant to an antibiotic that is supposed to kill it, then that bacteria can spread really quickly. That's why antibiotic-resistant bacteria can be really dangerous, because they can take advantage of that environmental situation, okay? So, since bacteria are asexual, there are no genders, there's no male and female bacteria, there's no sperm and egg production, so there is no true sexual reproduction, none, no sex. However, the point of sexual reproduction is creating variety, and as we've already mentioned, bacteria can create variety, and they can do that by means other than just mutating and getting new genes to pop up because of random mutations, okay? They can get new genes from other sources. So how can they do that if they don't engage in sexual reproduction? There is a process called conjugation. Conjugation. And to us, if we were just looking at two bacteria undergoing conjugation, we'd think they were having sex. It's what it looks like, but it's not. But they do kind of snuggle up next to each other, but they do not exchange even amounts of genetic material. Like in sexual reproduction in animals, half of the baby's chromosomes comes from their mother and half comes from their father. In the case of conjugation, they exchange, or one bacterium gives the other one bits of genetic material. I don't know if you remember what I mentioned earlier. If this is my bacteria and this is the main chromosome, many bacteria carry little small bonus bits of genetic material, of DNA, that are called plasmids, okay? Well, when bacteria undergo conjugation, they can swap these plasmids back and forth. It's not always equal. It's not a 50-50 kind of exchange, so it is not sex. It is just a random kind of exchange of a plasmid here or there. But it does help them get some variety. If there's a gene for being resistant to an antibiotic on that plasmid and another bacteria picks it up because of conjugation, okay, this goes to that one, then that's a huge, big improvement for that bacterium and all of its offspring. So, sometimes, here's all the background on plasmids that I've already told you about. So a plasmid is a small ring of DNA, and very often they do carry genes for antibiotic resistance, genes that work against those drugs found in the environment or given to you by your doctor that will kill bacteria. We often use the ability of bacteria not just to go through conjugation, but to do something else. Bacteria undergo also a process called transformation. Okay? And in transformation, bacteria just pick up plasmids from their environment. Maybe some bacteria died and the body decayed and left plasmids hanging around in the environment. So you don't have to go through conjugation to get a plasmid from another bacterium. A bacterium could just pick one up from its environment. Human beings, we scientists, use this property of bacteria to often get genes we want them to have into a bacterium. For example, if you remember, I told you in AP Biology, we make bacteria glow in the dark. This is how we do it. We put the glow-in-the-dark gene, we engineer it. If this is my plasmid, here, researchers cut the plasmid open, insert the glow-in-the-dark gene, and then stitch let the plasmid close back up again. And then when we put this plasmid in a bacterium, it will glow in the dark. It'll make the protein to make it glow in the dark. So in nature, bacteria can pick up random plasmids left behind by other bacteria, but we can also cause them to pick up plasmids that we want if we put them in a test tube with the plasmid. So it's a really useful thing. We're finding out more and more. We can make bacteria, if we put in a gene for human insulin in a plasmid and cause bacteria to pick it up, the bacteria can miraculously make human insulin. So it's really important. So picking up a plasmid from your environment is called transformation. All right. I'll stop there, let you take a break, and then you can pick up our talk of antibiotics in the next podcast. Don't want to stretch these out too long and make you bored. So I will see you next time. Take care.